Hi guys, my name is Karya Senkoylu and I'll present you the article called Magnolol inhibits the growth of non-small cell lung cancer via inhibiting microtubule polymerization. To be honest, the title successfully summarizes the whole point of the article. But I'd like to give you more detailed information by going through each section starting from the background to the discussion. Science background. What are microtubules? Microtubules are polymers of tubulin, which forms the mitotic spindle in mitosis and equally divides the parental cell into two daughter cells by chromosome separation. By targeting these cytoskeletal structures, their dynamics are disrupted, and therefore a lot of the diseases can be cured, such as cancer, by mitosis arrest of tumor cells. In this article, the inhibitory effect of magnolol on cancer cells microtubules is examined. The most common malignant tumor is lung cancer, and therefore the cause of related death in the world. Imagine, more than 1.5 million lung cancer cases are newly diagnosed every year, and 80% of which are non-small cell lung cancer. The morbidity is in a continuous increase due to pollution and unhealthy lifestyles like smoking, diet, etc. Therefore, there is an urgent need to identify drug targets to find new treatment methods, which is the aim of this article. And what exactly is magnolol? It's a compound isolated from the root and the stem of this magnolia flower shown in the picture. It has been recently concluded that it has anti-cancer properties which remain unknown for lung cancer. Therefore, this study is designed to investigate the efficacy of this anti-tumor activity against human NSCLC cells. The main method was to perform a microtubule polymerization assay to examine the effect of magnolol on lung cancer cells. Cell cycle analysis, mitotic index assay, cell proliferation assay, colony formation assay, Western blotting analysis of cell cycle regulators, annexin staining, and liver death viability staining were carried out to investigate the magnolol's inhibitory effect on proliferation and viability of tumor cells in vitro. Xenograft model of human A549 NSCLC tumor was used to determine the magnolol's efficacy in vivo. Afterwards, human NSCLC cells that are treated with magnolol were implanted subcutaneously to the male nude mice. 10 days later, mice were divided into two random groups. One group received magnolol intraperitoneally, while the control group was treated with DMSO. 20 days later, tumors were removed and images were taken. The growth rate of the tumor xenograft was evaluated by determining the tumor volume. To obtain the results, the tumor growth between two groups were compared statistically. And now we can move on to the results with their methods. Magnolol inhibits proliferation and colony formation of tumor cells. And how is it concluded? SRB assay is performed to examine this anti-proliferative effect of magnolol against tumor and normal bronchial epithelium cells. Then, its anti-proliferative effect on lung cancer cells is investigated by using colony formation assay. In accordance with these results, magnolol treatment led to significant inhibition on lung cancer colony formation compared to the controls. Taken together, these results demonstrate that it not only has a strong effect on the proliferation of tumor cells, but it is highly selective against these abnormal cells. The second result that we obtained was that magnolol induces cell cycle arrests of tumor cells. To prove the magnolol induced G2M phase arrests, the phase regulatory proteins were evaluated by Western blotting. As expected, the treatment of cancer cells with magnolol resulted in a dose-dependent downregulation of phosphorylated phase regulatory proteins. And magnolol disrupts microtubule organization in tumor cells. Its treatment decreases the polymerized tubulin in a dose-dependent manner. Immunofluorescent staining of the cellular microtubule at interphase and mitotic phase was performed to further examine its effects. In the absence of magnolol treatment, cells at interphase exhibited the normal microtubule organization, unlike the magnolol-treated cells, which showed more diffuse staining pattern throughout the cytoplasm. And we can see that magnolol provokes tumor cell apoptosis and autophagy. Cell apoptosis, when treated with magnolol, is approved by the annexin staining in flow concentration and time-dependent manners. Then, Western blotting analysis further indicated that magnolol treatment upregulated the protein levels of several apoptotic markers, such as cleaved caspase-9, cleaved PRP, and BAX. Lastly, magnolol inhibits the growth of A549 tumor xenograft. The growth of lung tumor was analyzed using tumor-injected mice. In this animal model, the tumor sizes of the magnolol-treated group were significantly reduced compared to the control alone. Magnolol also exhibited strong tumor suppression efficacy and low toxicity as it did in vitro cell culture system. Lastly, plants have been beneficial sources for pharmacotherapy worldwide from old. And in this study, magnolol has been used to show its potential in lung cancer regulation. Its inhibitor effect on NSCLC cell proliferation, growth, and colony formation, as well as tumor growth suppression, is successfully concluded with a series of experiments. Its antitumor efficacy is proved through the disruption of cellular polymerization of microtubule in lung cancer. Especially, NSCLC causes more deaths than any other types of cancer in both men and women. A lot of the cases may relapse and progress into late-stage metastatic disease. Even though chemotherapeutic agents are used, the upcoming drug resistance and their side effects inhibit their effectiveness. Therefore, it's necessary to develop new agents to overcome these hurdles. With this study, the decrease in cellular polymerized tubulin is shown to be under the control of magnolol exposure, which indicates its potential as a microtubule targeting agent. In addition to its anti-cancer effects, its acceptable toxicity holds great promise. Even so, the way that magnolol inhibits microtubule polymerization is not that clear. Does it bind to microtubule directly or through binding to other molecules? Therefore, there is still an urgent need to determine its pharmaceutical characteristics in more detail before it's clinically served for lung cancer treatment. Thank you so much for listening.